Am I audible, visible? Hello. Hi everyone, am I audible and visible to you all? Hi. Hi everyone, am I audible and visible to you all? Do let me know in the chat section. Happy morning, welcome to Unacademy NEET English. How are you? Hi. Hi kids. So am I perfectly audible and visible? Please let me know in the chat section, all of you. Okay. So let's start the new chapter today. So today we are going to start cell cycle and cell division. We are going to start from the basics of the chapter cell cycle and cell division. Right? Okay, so the chapter is interesting, the chapter is important, and the most important thing is that this particular chapter is linked to your class 12th chapters as well. If you master this chapter, then there will be no problem in the topics of chapter human reproduction, that is gametogenesis. Even in genetics, you need to relate this particular chapter. So kindly listen to me very carefully, kindly revise the topic properly, bache, right? And for this chapter, do make the notes as well. Okay. Orientation session will be there tomorrow, and I hope you all are aware that tomorrow we are going to start our first class for the Avengers batch, right? So if you have joined it, very good. If not, please be the part of Avengers batch, because before 26th of May, right, you people are going to get this batch at a price of 9599 rupees. So you guys can see here, bache, you can see any video, you just need to click, you just need to check the batch link here. Okay, so let me check. Okay, I don't know what's happening. So basically in the description box you will get the batch link. You need to click on that batch link, right? But unlock 20 and Avenger coupon code is applied, and you people will get this batch at a price of 9599 rupees, and afterwards this batch will be at a price of 14999. So be the part of this batch and join us from our first class. There should be no backlogs, bache. Be sincere for your NEET preparation, right? And let's start today's chapter. So today we are going to talk about the cell cycle and cell division. So before that, you have to tell me some basics of cell cycle and cell division. So tell me first thing, why do a cell divide? What is the reason behind the division? Yes, why do a cell divide? What is the reason? Thank you so much, Navansh. And guys, do hit the like button and do subscribe our channel. Please recommend this channel to your juniors as well, those who are in class 11th preparing for NEET, even those who are in class 10th, because time to time you people will get guidance that how to prepare for the NEET, right? What are the things that you people need to take care, the timetable, the books, everything we are going to provide you here on this channel. Okay, so do recommend this channel to your friends as well. Okay, very good, Mohammed. Excellent. So my question is, why do a cell divide? Growth, reproduction, okay, development to replace injury, good. But what else? What else? Basically, you can say that these are the benefits of cell division, right? Obviously, we are a multicellular body. Even if you talk about any unicellular organism, right? Even that unicellular organism is going to divide, isn't it? Isn't it? If I'm talking about the amoeba, obviously there comes a time when it will divide into two daughter cell. So we know that ultimately division is important. And if there will be no division, obviously the continuation of that cell life will not be there. And moreover, you are right, when we are getting some injuries, for the healing, the cell growth should be there, and it is there. Okay, it is there. So all that factors are there, no doubt, without any doubt, all these factors are there. We need cell growth, we need cell division for the growth as well, for the reproduction as well. But what is the thing that triggers that now there should be a cell division? What is the logic behind? Like all of a sudden, how can you say that today's cell has realized that I should divide? What is the reason behind? I'm getting one answer only. Thank you so much, Priyank. Quickly, everyone, just answer this question. It is very interesting class. And today I'm just going to talk about the basics. I'm not going to stretch this topic. I'll tell you what exactly is the cell cycle. Okay, so only basics we are going to revise, and this chapter is not so lengthy. So in 3 sessions, we will be able to finish it, and we will be able to finish it properly. So don't you people worry, we have the sufficient time, but you need to be sincere. You need to answer the questions properly. So tell me, what makes a cell divide? What makes a cell to divide? Yes, to multiply genetic material, not exactly, Priya. What else? Guys, answer it, answer it. Let's make it interactive. Anyone? See, there are 2 reasons, right? The very first reason is, let me mention it here. Why do cell divide? Right, this is what we are discussing right now. The reason is, bache, first is nucleocytoplasmic ratio, and the second thing is surface to volume ratio. These are the 2 reasons. One is nucleocytoplasmic ratio and another is surface to volume ratio. Now let me explain what exactly is this nucleocytoplasmic ratio. Nucleocytoplasmic ratio is also known as Kern-plasmic ratio, right? So basically nucleocytoplasmic means here we are talking about the nucleus as well, we are talking about the cytoplasm as well. So what is the story behind? Let me tell you first. So let's see, when a cell is small in size, its nucleus is going to be large. Right, when a cell is small in size, its nucleus is going to be large, isn't it? Right, when it is small in size, the nucleus is going to be large, and in comparison to that, cytoplasm is less. Right, bache? And now, this is a very general thing. A cell will continue the increase in its size. Okay, the cell will continue the increase in its size. Basically, it will show the cytoplasmic growth. Okay, it is going to show the cytoplasmic growth. So firstly, when the cell is small in size, at that time the nucleus is more and the cytoplasm is less. So you can say that nuclear to cytoplasmic ratio is high. Right, nuclear to cytoplasmic ratio is high, right, bache? Now when it will increase its size, the size of the nucleus will remain same, but the size of the cytoplasm, it is going to increase. So at that time, this nucleus cannot control everything properly, right? It is not that sufficient that it can, fulfill everything for that cell. So what is going to happen? The nucleocytoplasmic ratio will decrease, right? So this is also a sign, this is also a reason. Now it is the time to divide for the survival. Okay, it is the time to divide for the survival. Are you getting my point? Are you getting my point? The first thing. Now when you talk about the surface to volume ratio, right? But before that, so always remember, we always used to say, small size cells, they are more active in comparison to the large size cell. Do you remember this part? The cells having the small size, they are very active. If we compare the cell with the small size cell, are more active in comparison to the cell having large size. Do you know that? Right? So this is the first reason. Now the second is surface to volume ratio. See, bache, when a cell is small in size, obviously its surface area is more. Okay, when a cell is small in size, so at that time its surface area is more in comparison to the volume, right? So it can draw the things easily from the surrounding, right? Whatever is required for the cell, whatever is required for the metabolism, so it can draw it very easily. But when the size of the cell will increase, so what is going to happen at that point of time, bache? Now the volume is more and the surface area is less. Okay, at this point what is going to happen? The volume is more and the surface area is less. So cell will not be able to draw the essential things from the surrounding for its growth. right? For its growth, it will not be able to do that. So it is also a sign that now I should divide, right? I should divide into two so that I can survive. So these are the two things. These are the two things. That is why, right? That is why a cell undergo the division. Okay. Okay, so now there is a poll in the chat section, so all of you just answer that poll, and if you have anything, any query related to the Avengers batch, please fill that form which is given in the chat section. Okay. Okay. And if you are still thinking to join the Avengers batch, please join it quickly, because from tomorrow we are going to start our regular classes. Okay, if you will join it late, obviously it will be problematic for you, and maybe after some time we are not going to accept the enrollments as well, because it becomes difficult for us to complete your backlogs. We don't want you people to just watch the recording, so we want you guys to attend the live classes so that you can interact with us, so that you can clear your doubts. But for that you have to be the part of Avengers batch. Okay, so Avengers batch fees is already reduced. It is 9599 rupees, and it is for the complete course. Your 11th and the 12th will be completed. You will get the mock test, you will get the study material, you will get everything required to clear the NEET examination. Okay, so be the part of Avengers batch as soon as possible. Done, bache? So surface volume. Bache, Shiv, if you want to use the Unacademy platform, it is very easy. You will learn it. Right now, please do not spam, please focus here. So as I was saying, that surface to volume ratio, when a cell is small in size, its surface area is more, its volume is less. But when it increases, the volume will increase, the surface area, it will reduce. So it will not be able to draw the material from the surrounding for its growth. Okay, it will not be able to do that. Are you getting my point? So it is also a sign that now the cell should divide. So division is important for the growth as well. You know that, right? And even, you know, single cell things, they will become multicellular with the help of division only. I am getting an injury. The healing will be done with the help of division only, right? So division is important. Division is important. And how can we study that? We used to study it under the topic cell cycle. So the first thing is cell cycle, and then the cell division. And this is what we need to understand. Cell cycle and cell division. This is what we need to understand. And bache, it's a sequential event, right? It's not something random. It is going to be in sequence, right? There will be a sequence. It is a sequence basically. The cell is going to increase its size. The cytoplasmic growth will be there, right? And we will even see the DNA duplication. We will even see that how the DNA will get condensed, the chromosome formation, the division. So it is a multi-step process also, and it's a sequential event, one after another. The things are going to happen, the things are going to occur, and then your cell is going to divide. Okay, so this is what you need to understand. So now for that we are going to use the NCERT. Obviously we need to master it, so let's revise it from the NCERT only. So you know that the life starts from a single cell, and you know the name of that single cell, zygote, right bache? Life starts from a single cell, zygote. Even we, in our case, we are the multicellular body, right? We are the multicellular body, and you know that we are highly advanced organism. But in our case also, the life starts from zygote. So bache, when you talk about the zygote, na, zygote is a totipotent cell. What is zygote? It's a totipotent cell. So what is the meaning of totipotency? What is the meaning of totipotency? Totipotency is the ability. It is the capability, right? It is the ability of a cell, of a single cell, to grow into a complete organism. Right? It is the ability of a single cell to grow into a complete organism. This is totipotency. This word is important. This definition is important. You should note it down in your notes. What is the word totipotency? What is totipotency? The capability, the ability of a single cell to grow into a complete organism. The ability of a single cell to grow into a complete organism. So how will that organism? Obviously it is having the ability, it is having the genetic material, it is having all the information, but it needs to divide, it needs to become multicellular, then only it can form the complete body. So this is what you need to understand. right? Motive, to motivate, it is not because of stem cells. Stem cells are also having the property of totipotency. So the keyword here is totipotency. Okay, the keyword here is totipotency. So zygote is what? It's a totipotent cell. Okay, it's a totipotent cell, right bache? Right bache? So growth and reproduction, you know that they are the characteristics of a cell. Growth and reproduction, they are the characteristic features of a cell, right? All the living organism will undergo growth, they will reproduce. Okay, okay, so they will divide into two, you know that, right? Let's, one cell will divide into two. So this cell is going to be the parent cell, and here what do we have? We will be having the offsprings, right? Offsprings, and you can also call it as daughter cells, right? Daughter cells, offspring or daughter cell. So parent cell is there, parent cell will divide, parent cell will form daughter cell, or you can also use the word offspring. You can also use the word offspring. Are you getting it? Are you getting it? So newly formed daughter cell, they will grow, they will divide, they will give rise to a new cell population, right bache? And obviously it is possible because of the cell division. Are you getting it? Because of the cell division. So here you guys can see the cell cycle, the cell division, and this is what we need to understand in detail. Okay, so up to this part, if there is any confusion, do let me know, bache. If you guys are having any confusion, do let me know. Sure? It's same. See, progeny means the upcoming generations, right, Priya? When we use the word progeny, like there is one cell, that cell is dividing into two, then again it will divide into two again. So that is going to be its progeny, right? Its generation. When specifically I am mentioning the word parent cell is forming daughter cell, so at that point I am just talking about the two daughter cell that has formed from that parent cell. Okay, you can even use the word offspring for that. Progeny is like, you know, generation of that single cell. This is what I'm mentioning. Jennifer is asking one question. It is interesting. I would like to answer it. So, ma'am, cell division is reproduction. See, actually, when you talk about the unicellular organism, in the living word chapter also we discussed this. When you talk about the unicellular organism, now in the case of unicellular organism, growth and reproduction is same. Okay, growth and reproduction, these two things are same. Remember that mutually inclusive, mutually exclusive events. So it is kind of mutually inclusive event. In the case of unicellular organism, like say we have a single cell here. This cell is growing, this cell is growing, it is increasing its size, it's increasing its size, right? It's increasing its size. Okay, so there comes a time when it will suddenly divide into two. You know that the nucleus cytoplasmic ratio and the surface to volume ratio is disturbed, so it is going to divide. So here actually it was just growing, and by chance it is going to divide as well. Okay, okay, this is going to happen in this case. So here this single cell is growing, but again it is dividing into two, so it is also a kind of reproduction. So here in such organism, growth and reproduction is same, and that is the reason we do not consider it as a defining property, right? Growth and reproduction, they are not the defining properties. Remember, bache, the chapter living word, they are not the defining property. That is the reason. Okay, done, bache? So now here you guys can see, so cell division, very important. Now you know the importance of cell division. So what is going to happen here? The DNA replication, the cell growth will be there. The DNA will replicate, DNA will double its number, and cell is going to grow, bache. So what is going to happen here? All these processes, that is cell division, DNA replication and cell growth, have to take place in a coordinated way to ensure correct division and formation of progeny cell having intact genomes, right? Having intact genome. So what is going to happen? There will be the division, there will be the replication, there will be the growth, and all the things are in a coordinated manner so that a proper cell division can occur. And division is something very important, kids, right? The error should not be there in the division. Okay, the error should not be there in the division. Okay, so now what you need to know here, see, here we are using the word genome. Do you know the meaning of genome? Anyone in the class? Do you know the meaning of genome? What is the meaning of genome? Anyone in the class? Intact genome. No, the meaning of genome. Tell me. Entire set of genes. Anything else? Set of DNA. Anything else? If you check the definition of the genome, na, they mention one word, haploid. Haploid genetic content. Even you will find this word, haploid word will also be there. Many people talk about the word genome, right? Even, no doubt, we are talking about the genetic information of a cell. Without any doubt, here we are talking about the genetic information of a cell, but still, if you read it, even if you check it from the Google, you will find this word, the haploid genetic component of haploid genetic content of a cell. Can you tell me what is the meaning of that and why do we call it as haploid content? Yes. Why do we use the word haploid? Anyone in the class? See, today we are going to discuss about the interphase mainly. Okay. Today, what am I going to tell you? I'm going to tell you about the interphase mainly. So that is why we will just talk about the basics. The basics needs to be very clear. If your basics are clear, then the chapter mitosis and meiosis will be very easy for you. But you need to understand the basics first. Okay. Right. So what am I saying? That we use the word haploid content for the genome. The haploid genetic information of a cell. Haploid. Why is it so? See, I'll give you one example. I'll give you the example of humans. In the case of humans, we have 46 chromosomes, right? What do we have? We have 46 chromosomes. 46 chromosomes means 23 pairs, isn't it? 46 chromosomes means we have 23 pairs. So if I talk about the 23 pairs, so you know that the one chromosome is from mother and another chromosome is from father. We have the things in a pair now. One chromosome is from mother, the another chromosome is from father. So obviously here we are having some genes and that genes are going to be same. Let's say here I have the gene for height. So on father's chromosome also I will have the gene for height only. I will have the gene for height only, right? Although one is from mother, another is from father, it is the allele that varies, right? But overall the gene is same. So in haploid content also you are going to get the same information. You are going to get the same information, same genetic information is there. You will get to know about the position of the genes, everything is same. It's just that, it's just the allele that varies. Okay, it's just the allele that varies. Are you getting it? Are you getting it? So that is why we used to mention haploid pair. Like out of these 46 chromosome, if you only consider 23 chromosome, you will get to know about all the genes present in that particular individual. Okay, you will get to know about all the genes present in that particular individual. That is the reason behind. Okay, so now here you can see the sequence of events by which a cell duplicates its genome, synthesize other constituents of cell, and then eventually divide into two daughter cells. It is the cell cycle. So what is it? It's a sequence of events. One after another the things are occurring. Cell is going to grow, the DNA will duplicate, and finally it is the cell cycle. Cell cycle. And bache, this is something very important. When you people talk about the cell growth, cell is growing, right? What are we discussing? We are talking about the cytoplasmic increase, right? What are we talking? We are talking about the cytoplasmic increase. The growth is there in the terms of cytoplasmic increase. The cytoplasm is increasing basically, right? Like here you are having a small sized cell, so obviously it will grow, it will become large size. So ultimately, why is it possible? There is the cytoplasmic growth. Cytoplasmic content is increasing. Are you getting my point? So we will always see the cytoplasmic growth, right? We are always going to see this cytoplasmic growth. So although cell growth in terms of cytoplasmic increase, it's a continuous process. What is it? It's a continuous process. Continuously you will see it. But DNA synthesis will only occur in a specific stage. So if anyone is asking you a question that in the cell growth, what is going to happen? The DNA will replicate again and again. The DNA is going to replicate again and again. Is it so? Right? Right. So no, it is not like that, because DNA synthesis will only occur. Right? The DNA synthesis will only occur. Just a minute, the DNA synthesis will only occur during one specific stage, and it is a repeated, right? It is a repeated NEET question. It's a repeated NEET question. Okay? Got it, everyone? Yes? So this is what we need to understand now. Now we need to talk about the phases of the cell cycle, and when we discuss the phases of the cell cycle, obviously all your doubts will be clear. So what do we have here in the phases of cell cycle? Just a minute. We have two things, the interphase and the M phase. When we talk about, when we talk about the phases of cell cycle, we have the interphase and the M phase. One is the interphase, another is the M phase. This is what you need to remember. Interphase, M phase, interphase, M phase. This is what you need to remember. And moreover, bache, when you talk about the interphase is a phase in between two successive M phase. Interphase is a phase in between, two successive M phase. This is what you need to remember. So what are we talking? We are talking about the phases of cell cycle. One is the interphase, another is the M phase. One is the interphase, another is the M phase. So now I am going to explain you this part, okay? Just a minute. The point is interphase and M phase. Interphase and M phase. Now, bache, when you talk about the interphase is even known as I phase. It is even known as invisible phase, right? Even it is known as resting stage, and even we used to call it as energetic phase. So it is the interphase, I phase, resting phase, invisible phase, energetic phase, even the preparatory phase, right bache? Even the preparatory phase. There are so many names for this interphase. And when you talk about the M phase, it is the actual division phase. Okay, there was a question in the NEET 2023 exam, M phase. M here is mitosis. Even we are going to discuss the cell division, meiosis also, right? But M here is defining the actual cell division phase, right? It is defining the actual cell division phase. So what do we have? We have interphase, we have M phase. Interphase, M phase. So interphase, I phase, invisible phase, resting phase, energetic phase, preparatory phase. And when it comes to the M phase, it is the actual cell division phase. What is it? It is the actual cell division phase. Are you getting my point? It is the actual cell division phase. Okay, the division is going to occur in this phase. Now, what is the point that you people need to understand? Why do we say that interphase is the resting stage, bache? Actually, when scientists initially, when they observed the interphase nucleus under the microscope, so at that time they were like, the cell is just taking a rest. It is doing nothing because changes were not so visible. Okay, initially what happened, the scientists, they were visualizing the things under the microscope. They were observing the things under the microscope. So they were observing the interphase nucleus. So at that time they were like, that interphase nucleus is not showing anything. There is no activity. The genetic material is, it is tangled. It is just placed like that only. Okay, it is just placed like that only there. These are the things. Are you getting my point? These are the things. So they observed that in the interphase, cell is doing nothing. But later on with further studies, we got to know that in the interphase, actually it is the cell is very active at that time. Cell is doing everything possible for this M phase. Cell is preparing itself for this M phase because this M phase should be error free. Okay, this M phase should be error free. Are you getting my point? So cell is doing everything possible here in this I phase so that it can have an error-free cell division phase. Okay, I always give the example of the NEET preparation, bache. Now, when you people talk about NEET, right, you guys are preparing for this particular exam. So for this particular exam, you used to give your 2 years initially, your class 11th, class 12th. If required, you take a drop, you even prepare for 3 years, you even prepare for 4 years, isn't it? You even prepare for 2 years, 3 years, 4 years. Why? You are preparing for an exam, and that exam is just for 3 hours 20 minutes. The duration of exam is 3 hours 20 minutes, and you give your 2 years, 3 years, 4 years for this exam, because you want to make sure that here on the exam day you make no mistake, isn't it? Isn't it? So if I see from another people, if I'll tell you the perspective of other people, so according to them you are doing nothing at this time, isn't it? Right? No one is going to understand that you are preparing for the NEET exam. You have to complete the syllabus, you have to complete the syllabus on time, you have to give the mock test, you have to make the notes, you have to revise the things again and again, you have to make sure that you are covering the entire syllabus, or you are revising it many times, isn't it? Isn't it? So for others, if I see from another people perspective, so they'll be like, they are just studying, they are just preparing for the NEET examination, they are doing nothing. Like if anyone asks it from your parents, so parents will also be like, they are just preparing. My kid used to sit in the home, they used to see the books for 3 hours or 4 hours, I don't know what are they doing, right? We will only decide when the result will come. Right now we cannot comment, right? What are they doing? And people will keep on asking you, and you will be like, I am preparing. But according to them, they are just preparing. But you know that this phase is so difficult, isn't it? So here in this phase you have to make sure that you are utilizing your time properly, you are not wasting your time, isn't it? You are utilizing your time properly, you are not wasting your time, isn't it? Isn't it? But you know how important this phase is. So same is the case here in the phase of cell cycle. Here in the phases of cell cycle, the interphase is that phase, right? We are calling it as resting phase, we are calling it as invisible phase, I phase, energetic phase, but actually here in this phase you are doing everything possible which is required for the cell cycle and cell division. Like cell division is going to take a lot of energy, right? Cell division is going to take a lot of energy, right? Are you getting my point? A lot of energy is required for this phase. So obviously all that energy, all that ATP will be synthesized here in this interface. Here in the interphase we need to even divide, we need to even duplicate our DNA. So all that things that we need to, right, that we need to duplicate our DNA, we are going to make it in interphase. So actually we are doing so many things to make this exam error free. Same is the case here. That is how you have to relate it. So can I say that in the interphase the cell is very active, the changes are not visible, but again the cell is very active, isn't it? Can I say so? Can I say so? Yes. So I think now you are not going to forget this, right? So you have to relate it with your NEET preparation. You have to relate it with your NEET preparation, right, bache? So when we talk about the interphase, so now I'll tell you about the interphase. Interphase, the word is what? The word is interphase. So here we have G1 phase, I'll explain it. We have S phase and we have G2 phase. We have G1 phase, we have S phase, we have G2 phase. So when it is the G1 phase, so what is it? It is the growth 1 phase, yeah, first growth phase, okay, and then it is the gap, it is the gap 1 phase, and even you used to call it as post mitotic phase. I'll explain you the reason behind, right? We even call it as post mitotic phase. What is it? It is the post mitotic phase. Now when you talk about the G2, it is again the growth 2 phase, the gap 2 phase, or you can say that second gap phase, and it is known as pre-mitotic phase. What is it? It is known as pre-mitotic phase, isn't it? Isn't it? Now here we have this S. So what is this S? S is the synthesis phase. It is the synthesis phase. So first of all you need to remember this sequence, then I'll explain you the everything. G1 S G2, G1 S G2, G1 S G2, and here we have one more thing, that is G0 phase. It is basically the cell arrest stage. We will discuss everything, you just need to listen to me carefully now. Cell arrest stage, and we can also call it as quiescent stage, right? We can also call it as quiescent stage. What is it? It is the quiescent stage. So G0 phase, cell arrest stage, the quiescent stage. Okay, are you getting my point? The quiescent stage. quiescent stage. This is what you need to understand. Okay, okay. Now in the NCERT there is one line, and I think you all should remember it. Then you will be able to relate the things in a better way. See, right now I'm not telling you what is happening in G1 and G2. We will discuss it, but before that, just listen to me carefully. In the NCERT there is one thing, that interphase is the phase between, it is the phase between two successive means one after another, between two successive M phase. It is given now. What is interphase? It is the phase between two successive M phase. What is it? It is the phase between two successive M phase, right? So M phase, I phase, M phase. That's how I can write it, yes or no? M phase, I phase, M phase, isn't it? I can write it like this. Yes, in M phase, I phase, M phase. Fazan, if they are better, then what are you doing here? Yes, what are you doing here? Done. So this is the meaning of this line. The interphase is the phase between two successive M phase. Two successive means two one after another. M phase is there. It is the phase. Like, let's say this is a cell. Cell is dividing. We will be having two daughter cell. Then these two daughter cells will be in that resting phase, and again they will divide. So obviously I can say that, right? I can say that M phase, I phase, M phase, M phase, I phase, M phase. Isn't it? Isn't it? है ना? है ना? So now in the I1, in the I phase, G1 is there, S is there, G2 is there. Okay, and when it comes to the M phase, when it comes to the division phase, when it comes to the mitosis or even in meiosis, so what is going to happen first? Firstly, there will be the nuclear division. There will be the nuclear division, and then there will be the cytoplasmic division. There will be the nuclear division, there will be the cytoplasmic division, right? Nuclear division means yes, karyokinesis, cytoplasmic division, cytokinesis, right? It is cytokinesis. Are you getting my point? Karyokinesis and cytokinesis, karyokinesis and cytokinesis. So when you talk about the M phase, firstly it is the nucleus that will divide, right? And then there will be the cytoplasm that is going to divide, right? It is the nucleus that will divide, and then comes the cytoplasm which will divide. Nuclear division, cytoplasmic division, nuclear division, cytoplasmic division. This is the sequence. So this is what you need to note down in your notes. This is what you need to mention in your notes. M phase, I phase, M phase, M phase, I phase, M phase. So M phase, nuclear division, cytokinesis, karyokinesis, cytokinesis, then G1, S and G2. Now just look at it, Jennifer. So can I not say that when we talk about the G1 phase, when we talk about the G1 phase is after mitosis. So can we not say that it is post-mitotic phase? Yes or no? It is the post-mitotic phase. We can say so, isn't it? And now when you talk about the G2, again it is before mitosis, so obviously we can say that pre-mitotic phase. What is it? It is the pre-mitotic phase, isn't it? And S is the, right? S is the synthesis only. S is the synthesis phase only, isn't it? Isn't it? Are you getting it? Right now we are more focused on I phase, but I am explaining you each and everything which can be asked in the paper. And you should know, but your concept should be very clear. See, the NEET exam is easy, I'm telling you. These days the biology is very, very, very easy. If you are thinking I will just read the NCERT, I will read it many times, I'll solve the question, okay, you will get good marks. But what about that knowledge, the information, that concept? Right? If you will understand it, see now, I know the things, so there is no need to revise it again and again. I know it, okay, I have understood it, right? So this is what you need to take care. Done, bache? So you need to revise it again and again, and you need to understand the topic. So now tell me, this flowchart is easy. With the help of it, you can remember this line, right? The question can come from this part, and post-mitotic and pre-mitotic phase will also be done, right? So I phase, the most energetic phase, the preparatory phase, the cell is preparing itself for the very important thing, that is division. So now, can I not say that, if I'm taking the example of this particular part, so can I not say that a cell is going to spend more than 95% of its lifespan in the interphase? Can I not say that? There is no need to cram this particular part, Arya. I have no need to cram this particular part, right? If I know this example, if I can relate it here, so I can say that a cell is going to spend more than 95% of its life cycle in the interphase, and rest is the division only. Like if I talk about the human cell, right? So 24 hours, in the 24 hours there will be the division. Within 24 hours the cell will divide once, right? Within 24 hours the cell will divide once. So you can say that the cell division is just of 1 hour. Are you getting it? The cell division is just for 1 hour. Rest 23 hours are of the interphase, right? They are of the interphase. So in the case of yeast, 90 minutes it is. In the case of E. coli, it is going to be near about 20 minutes. So in different cell, this duration will vary, but ultimately the logic behind is that it is the interphase where cell is going to spend maximum time, okay? Where cell is going to spend maximum time, okay? So is that clear, bache? Is that clear? So now just look at it, revise it from NCERT. So a typical eukaryotic cell is illustrated by a human cell in a culture. The cell will divide once in approximately every 24 hours, right? In every 24 hours it will divide once, okay? So as I said, the duration of cell cycle will vary. So yeast, it is for 90 minutes, and here you can see interphase and M phase. So M phase represent the phase when actual cell division is going on, right? But here interphase is the phase between two successive M phases. So is that clear? It is the phase in between two successive M phases. Is that clear, bache? So it is significant to note that in the 24-hour average duration of cell cycle, cell division will last for only about an hour. The interphase lasts for more than 95% of duration of cell cycle. So up to this part, I hope everything is clear to you. Okay, okay, done, bache? Done. So this is the diagram from NCERT. I will explain it, but before that, let's talk about the interphase. Done. Interphase. So okay. So G1 phase. But when it comes to the G1 phase, it is the most variable phase, right? It is the most variable phase. Can you tell me the meaning of this line? Why are we saying that? It is the most variable phase. Now just look at this particular diagram. Here you people can see we have G1, we have S phase, we have G2 phase, then comes the M phase. Now when you talk about the M phase, bache, firstly there will be the nuclear division, then there will be the cytoplasmic division. Okay, when you talk about the M phase, see this portion is karyokinesis, nuclear division it is, and this portion is what? Cytokinesis, cytoplasmic division. Are you getting my point? Cytoplasmic division. Are you getting my point? And after that, cell will divide into two. See, this is a single cell. It will undergo interphase. After all that phases, you will get two daughter cells. This is the thing basically, G1S, G2, and then comes the karyokinesis and cytokinesis. G1S, G2, karyokinesis, cytokinesis. Now, bache, what I said, that G1 is the most variable phase, right? What is it? It is the most variable phase, right? Now, what is the meaning of it? It means, if I talk about the duration of G1, it can vary. You cannot say that in all the cells, the G1 will last for, 10 hours or 12 hours or 13 hours. No. Its duration can vary, bache, right? Its cell duration is, its time span is not fixed, right? Let's say I have this cell and this cell used to divide very quickly, right? I have a cell this cell used to divide very quickly, right? In this the division is very quick. So if division is very quick, obviously G1 phase will be of short duration, right? It will be of short duration. So the phase, the duration, this timing, it changes. And when you talk about the G1, it is the most variable phase, and out of all the phases of the cell cycle, it is even the longest. Phase of the interphase. Okay, it is the most variable phase, even it is the longest phase. Money, even it is the longest phase. This is what you people need to remember. I'll quote one more example here, right? But all of you know about the zygote. All of you know about the zygote. I told you it is a totipotent cell. It is a cell which will undergo division, right? It is a cell which is capable of growing into a complete organism, isn't it? So when you talk about this zygote, right? So zygote initially it divides very quickly. Very quickly it is going to form a multicellular structure. So at this point, the cell division is very quick. We use the word cleavage for that, right? We use the word cleavage for that. Cell division is very quick. Cleavage word is used because at this point of time, here it is not having the G1 phase, it is not having the G2 phase. So that is why I am mentioning that this is the most variable phase and the longest phase, and this is what you need to take care. Okay, this is what you people need to remember. Okay, done. So that is why I am mentioning it here. So now what is going to happen in the G1 phase? Of course the protein synthesis, right? The protein synthesis. Proteins means enzymes. Mostly enzymes are proteins, so they will be synthesized here. Even RNA formation will be there. And moreover, remember this thing, when a cell is in G1, it is preparing everything required for S phase. When it is in S, okay, when a cell is in G1, it is doing everything which is required for the S phase. Right, it is doing everything which is required for the S phase. So in the G1 phase, the most variable phase, the protein synthesis will occur, the RNA formation will be there, and mostly organelles, right? Most of the organelles will duplicate in this phase. Okay, most of the organelles duplicate in this phase. Okay, they will duplicate its content. They are going to duplicate its content. Okay, because ultimately when a cell will divide into two, so equal number of organelles will also be distributed, equal number of content will be distributed. So obviously the duplication of the organelle, most of the organelles will duplicate in this phase. They are going to double their content. They are going to double their content. Are you getting my point? So G1, most variable, protein synthesis will occur, RNA formation will be there, and obviously active ATP synthesis will also be there in this phase. Okay. Okay. ATP synthesis. Of course, ATP synthesis will also be there in this phase. Now, after G1 phase, there will be the S phase. After G1 phase, what do we have? We have the S phase. S means synthesis. So, it is the only phase of interphase where the DNA replication will occur. It is the favorite question of your NTA, right? They always used to ask the question from this part. So S phase is synthesis. What is going to happen in S phase? The DNA replication will occur, the DNA duplication will occur, the DNA will duplicate into 2. The DNA will duplicate into 2. Okay. Okay. Done? Is that clear? So, most variable phase, protein synthesis, RNA formation, organelles are going to duplicate their content in this phase. The active ATP synthesis will be there. Now, I am going to relate it with this S phase. Now, as I said, S phase is the phase where the duplication of DNA, or you can say that DNA replication will occur. What is going to happen in this phase? The DNA replication will occur, and even, but just a minute, number of chromosome will remain same. Now this is something important that you need to understand. Number of chromosome will remain same, right? So if I denote the DNA content as 2C, so after S phase it will become 4C. 2C, it is basically denoting the DNA content in a cell. 2C is denoting the, right, DNA content of a cell. So after S phase, 2C will become 4C. 2C will become 4C. But the main point that you need to remember here is the number of chromosome will remain same. So bache, I'll quote one example here because this is the part where many students they get confused. Okay, these are the picogram values bache. You can take in different ways. You can use any other unit as well, dino bache. Okay? So now the point here is, see bache, when a cell is in the resting stage, actually what is happening, if you look at its nucleus, the DNA is not condensed. It is not in the form of chromosome. Right? When you look at the nucleus of a cell, which is an interface, the DNA is not there in the form of the chromosome. It is not intact. It is not intact. It is just like this. It is tangled. It is tangled. It is placed here in the nucleus. Okay. But now, because I want to give you one example, right? I want to give you one example. So I will consider the intact chromosome. Okay, I am going to consider the intact chromosome. So here let's see, this is a cell. Here in this cell, let's say number of chromosome is 4. Okay, let's consider the example of this cell. So here the number of chromosome is 4. What is the number of chromosome here? The number of chromosome is 4. Let's consider one example. Okay, okay. So right now we are showing the DNA is placed like this. It is tangled. It is tangled. It is not compactly arranged. It is not compact. It is not in that chromosome form. But you imagine that it is in chromosome form. So bache, this will be the structure here. Many times students, they are not able to understand, right? They think that only this structure is chromosome. Many times students, they just consider that only this structure is chromosome. They are not aware of it. But it is also a chromosome. It is not chromatid. It is chromosome. It is the chromosome before S phase. It is a monad chromosome. It is a chromosome in which DNA content is not doubled, in which DNA duplication is not there. Are you getting my point? So if I'm saying that we have the example of a cell where the number of chromosome is 4. So before S phase, you can show it. You will show it like this. Okay, you will show it like this. Right? So it is the monad chromosome. Single arm is there. Only single arm is there because DNA duplication is not there yet. DNA duplication is not there yet. So how many chromosomes are there? 1, 2, 3 and 4. Isn't it? 1, 2, 3 and 4. How many chromosomes are there? 1, 2, 3 and 4. Isn't it? Isn't it? Yes? Of course. Of course. Now, after S phase, as I said, in S phase, what will happen? The 2C will become 4C, means DNA replication will occur. The DNA content will get doubled. So, here in this chromosome, ultimately, so it is the DNA. Right? Ultimately, it is what? It is the DNA, isn't it? Ultimately, it is what? It is the DNA, isn't it? Yes or no? Yes or no? Quick, quick, quick. Yes or no? Of course, yes. So, after S phase, what will happen? The DNA content will get doubled. So, the chromosome will become like this. There will be the 2 arms. Now, you will be having the sister chromatids. Right? Now, we will be having the sister chromatids. So, see, 2C will become 4C. So let's say here if the DNA was 100 grams. So after S phase, it is going to be 200 grams. Right now I'm using the unit gram. Just take it like this. Right? So before S phase, if it is 100 grams, so after S phase, it is going to be 200 grams. It is going to be 200 grams. So before S phase, the DNA, the genetic material is just like that. Right, the arms are not doubled. In the case of chromosome, the DNA content is not doubled, but after S phase, it will be doubled. There will be two arms, right? The 2 chromatids will be there. So all I can say is the DNA amount is getting doubled. But if I talk about the number of chromosome, again the number of chromosome is same. It is going to be 4, right? It is going to be 4, right? So now you got the logic behind. Yes, Arya? It is important, bache. Okay, done. G bache Pratyusha. Okay, right? So always remember, it is the DNA content that will get doubled, but the number of chromosome will remain same. So 2C will become 4C, but when you talk about the number of chromosome, even after S phase, the number of chromosome will be same, that is 4. Okay, so along with that, in the S phase, what is going to happen, bache? In the S phase, there will be the duplication of centriole. Do you remember the structure centriole? I have explained it in last class. Duplication of centriole. Centriole is a non-membrane bound organelle which is present in the animal cell. So that centrioles will also get duplicated here in the S phase, right? In the S phase. And moreover, histone synthesis. You know, histone protein, the basic proteins, the positively charged protein. So DNA will coil over these positively charged proteins. So histone proteins will also get synthesized here in this S phase. Histone proteins will also get synthesized here in this S phase. Okay, and now I will relate the things in the G1 phase. Now you know after G1 there will be the S phase. So in the S phase, DNA duplication will be there. So all the enzymes which are required for the duplication of DNA, right, we have studied, we will study the process in detail in molecular basis of inheritance. So we need some enzymes, we need some DNA polymerases, we need the ligases. So all that enzymes which are required for the duplication of DNA, they will be synthesized in G1 phase. So that is why we used to say it is a sequence of events, right? It is a sequence of event. Are you getting my point? Sequence of events. So in G1, what is happening? What is happening here in the G1, bache? In the G1, the protein synthesis will be there. The active enzyme synthesis will be there. For the S phase, in the DNA duplication, even we need the ATP. So all that ATP synthesis, the active ATP synthesis will be there, right? Even in S phase, you know that for the DNA duplication, the nucleotides are required. So all that will synthesize here in the G1 phase. Are you getting my point? They will be synthesized in the G1 phase. Then comes the S phase. Then comes the S phase. Clear, bache? Clear, bache? So the next phase, and the next phase that we have is, it is the G2 phase. It is the G2 phase. What is it? G2, gap 2, or you can say that pre-mitotic phase. Now you know why do we call it as pre-mitotic phase? Okay, why do we call it as pre-mitotic phase? Here also, bache, the RNA synthesis, right, the protein synthesis, all that things are going to occur. Even the ATP synthesis will also be there in this particular phase. Okay, RNA synthesis, protein synthesis, all that things are going to take place in this G2 phase. Okay, and the microtubules, the tubulin protein, right, you know na, microtubules used to form the spindle apparatus. We need the spindle apparatus in M phase. Remember, Samiksha, we need the spindle apparatus in the M phase. In the M phase, for the division of that cell, for the separation of that chromosome, we need spindle apparatus. Spindle apparatus is made up of microtubules. Microtubules are made up of tubulin protein. So tubulin protein synthesis will also take place here in this G2 phase. Okay, tubulin protein synthesis will also take place in this G2 phase, right? So this is how you have to remember it, right? So this is all about the interphase. This is all about the stages of the interphase. Okay, okay. Now, bache, there is one more point. Here we used to write one thing, na, that is G0 phase. I told you that it is the cell, it is the quiescent stage. It is the cell arrest stage, quiescent stage and the cell arrest stage. This is what I have mentioned for the G0 phase, isn't it? For the G0 phase, this is what I have mentioned. But yeah, you know, many cells, they just grow, they do not divide. Okay, many cells, they just grow, they do not divide. Like if I take the example of heart cell, if I take the example of your neurons, the longest cell of human body, in their case, no doubt they are going to show the cytoplasmic growth, right? They are going to show the cytoplasmic growth, but in their case, you will not see active cell division. And even there are some other cells also, bache, in which, you know, they do not want to grow they do not want to divide immediately. They will just increase their size. Like let's say here we have one cell. This cell is growing. This cell is growing. It is showing the cytoplasmic growth, but it is not divided into two. So in that scenario, what is going to happen? The cell will leave the G1 and it will enter in G0 phase. Like after cell division, that cell will enter in G1. If it will not get go ahead signal that now I should go and I should divide, I should go and I should divide, right? So if it will not get it, so it will enter in this stage G0 phase. Okay, it will enter in this stage, that is your G0 phase, right? It will enter in G0 phase. So here in G0 phase, the cell division will not occur. The cell division is not there, but bache, cell is metabolically active. Although we used to say it is a cell arrest stage, but here you cannot say that metabolism is not there, right? Right, we cannot say that metabolism is not there, right? Cell division will not occur, but the metabolism is there. So you can say that it is a phase in which it is an inactive stage. Overall, it is an inactive stage because division is not occurring, but cell is metabolically active, right? The cell is metabolically active. Are you getting my point? Cell is metabolically active. But when you talk about metabolism, what is metabolism? Metabolism is all the chemical reaction which are going on in a cell, right? Metabolism is all the chemical reaction which are going on in a cell, and metabolism is a defining property of living organism. If something is alive, right, it means the chemical reactions are going on. Okay, so metabolism mein we have two things. One thing is anabolism, another thing is catabolism. One thing is anabolism, another thing is catabolism. Ana, see, kata means cut, means breakdown. That is how you can remember it. Kata means cut, means breakdown. Okay, you are going to break something, you are going to cut something, and anabolism is going to be build up. So metabolism means sometimes some things will break down, there will be the breakdown of some things, there will be the build up of something. So collectively it is the metabolism. So if anything is live, it means metabolism is going on. If anything is live, it means metabolism is going on, right? It means metabolism is going on. Are you getting my point? Yes, are you getting my point? Right? So although if a cell is in G0 phase, it is not the dividing, the division is not there, but cell is metabolically active. It means the metabolism, the chemical reaction will always be there, and it is important for the survival. Okay, it is important for the survival. Okay, bache, and moreover there are some checkpoints. The topic of checkpoints is not given in NCERT, but it is important. Okay, it is important. Just a minute. See, here in G1 now you have one restriction point. If I talk about the G1 bache, there is one restriction point. When a cell, it enters the G1, if it crosses this restriction point, right? Actually the checkpoints are, just like the barricades that we used to get on roads, right? The people, obviously the forces, they check everything, they check our vehicles, they check everything. Barricades used to be there, na? Same is the case here. Because cell division is something very, very, very important. It should be error free. If there will be any error at the level of genes, at the level of DNA, at the level of chromosome, so it will result in abnormalities. It is going to result in abnormalities. So this process should be error free. It is going to be error, it should be error free. So here in G1, firstly, the very first thing here we have is this checkpoint is restriction point. What is it? It is the restriction point. From G1, the cell will enter in S. So if everything is good to go, so if a cell crosses this particular restriction point, then for sure it will complete the cell division. I told you in G1, there are two scenarios. If a cell is going to divide, then it has to cross this restriction point. It has to cross this checkpoint, right? And if it crosses this checkpoint, then definitely it will complete the circle. It will complete the cell division. And if it is not going to divide, it will leave G1, it will go in G0. It will leave G1, it will go in G0, right? Right? It will go there. And moreover, in S phase, the DNA synthesis and everything is there. Even it should be error free. And in the cell, there are many genes, there are many, cyclin dependent kinases, some other enzymes are there which are going to take care of it. So they are not required in detail, but whatever is required, I'm telling you. So one checkpoint is this, right? So if a cell is crossing this main checkpoint, G1 to S wala checkpoint, so for sure it is going to finish the cell cycle. It is going to divide, the first thing. Second is after S phase, it will enter in G2 phase, and then it will go in M. So from G2 to M, there is another checkpoint, right? right? So here this checkpoint will make sure that DNA is proper, the division is proper, and if there is any abnormality, they are going to stop the cell cycle. Okay, right? It is going to stop the cell cycle. So this is what you need to revise, right bache? So this is all about the cell cycle. So now I think you can understand this diagram, G1S, G2. Now this is the karyokinesis, cytokinesis. So in detail we will discuss it in next class. Here you have the two daughter cell. So after G1, G0, right? And I told you about the G0 as well. So let's revise it from NCERT. Let's solve few questions and then we will end our class. Okay, then we are going to end our class. So here you can see that M phase starts with the nuclear division, that is your karyokinesis, and then comes the division of cytoplasm, cytokinesis. But in interphase, the resting phase it is. So the cell is preparing for division by undergoing both cell growth and DNA replication. So what can we say that in interphase what is happening? The growth of cell and the DNA replication. The growth of cell and the DNA replication. So here you people can see G1S, G2. G1S, G2. Okay, G1S, G2. So G1 is the interval between mitosis and initiation of DNA replication. We can say that, right? There is the M phase, then there is the I phase, G1S, G2, and then again there is the M phase. So of course we can say that G1 phase is the interval. It is that gap in between M phase and S phase, in between M phase and S phase. So it is interval between mitosis and initiation of DNA replication. Okay, bache? Okay, so during G1 what is going to happen? During G1 the cell is metabolically active, it is growing, but it will not replicate its DNA. But in S phase the DNA replication will be there. So if DNA, we are denoting the DNA with 2C, so it will increase to 4C, but there will be no change in the chromosome number. If the cell is diploid, it will stay diploid, right? And in animal cell during the S phase what will happen, bache? DNA replication begins in the nucleus and the centriole also duplicate in the cytoplasm. And in G2 again the protein synthesis and everything will occur. So bache, this is given in your NCERT. Just read it. How do plants and animals, and this is what you need to answer. You have to answer it in the comment section. This is your homework, right? I'm not able to see your answers in the comment section. Only few students are answering the questions in the comment section. So please do it. It is important. Kindly do it. Okay, so how do animals and plants continue to grow all their lives? Do all cells in a plant divide all the time? So these are the questions that you need to answer, and I want to see the answer in the comment section right after the class. Okay, right after the class, this is what you need to mention. And here you people can see about the quiescent stage also, we have discussed it. So this line is very important, that cell is metabolically active, but although there is no proliferation. Proliferation means a division. Cell is metabolically active, but it will not divide into two. It will not divide into two. Clear, bache? Clear? So it is the inactive stage. So quiescent stage is inactive, but cell is metabolically active. Metabolism is going on. Okay, metabolism is going on. So rest bache about the M phase and about the about the mitosis and meiosis we will discuss in next class. And this is another question that you people need to answer. So you have studied mitosis in onion root. They are saying 16 chromosomes, right? 16 chromosomes will be there at the G1 phase. So after S phase and after M phase, what will be the chromosome number? You know that number of chromosome will not change. So if in G1 it's S, in S phase also it will be S, in G2 also it will be 16. Like if in G1 the number of chromosome is 16, after S, after G2 it is going to be 16, because there is no change. There is no change in what? In the chromosome number. And if you talk about the DNA content, let's say in G1 it is 2C, after S phase, in S phase it will become 4C, in G2 also it will be 4C, and after M phase, right, the two daughter cells that we are going to get, they will also have 2C and 2C. Okay, ma'am, in the cell present in G0 have double the, no, why bache Gokul? I told you now that if they will get go ahead signal, they will cross that restriction point, then they will enter in the S phase. But if they are not going to go for that division, so obviously it will enter in the quiescent stage. It will leave it early. Only the cytoplasmic growth will be there. Okay, only the cytoplasmic growth will be there. So bache, this is all about your cell cycle, and everything important we have already covered. So now what you have to do, okay, right, if you have joined the Avengers batch, then good. Otherwise you can be the part of this batch, because we are going to start the classes from, yes, we are going to start the classes from tomorrow. So tomorrow, like at 12, you will be having your first biology class. Okay, you will be having your first biology class. So the schedule for biology class is Monday, Tuesday, Thursday, Friday. Okay, so at 12 I will take your class, 12 to 1:30 I'm going to take your classes, right? And the physics class will be there in the morning. But because of some reason these days, like we are going to start the batch from tomorrow, so you will have your class on Thursday, Friday, even on Saturday. Done, bache? Done. So I hope you like the session. So do subscribe our channel, bache, and please make the notes, make the proper notes, and whatever I'm telling you now, that mention it in the comment section. Do mention it. In this way you will revise, you will always remember it, and do practice the question based on this part, that what will be the DNA content in G1, what will be the DNA content in G2. Revise these things. Okay, revise these things. Okay.